Hello everyone, myself Prerana Pawar, studying in final year of B Pharmacy from Shivajirao West Only College of Pharmacy, Asangaon. Today I am going to present on topic wound healing and anti-inflammatory activities of Jatropha CURCAS plant, guided by Assistant Professor Nikita Pagare ma'am. We are going to discuss about introduction, wound healing, inflammation, Jatropha CURCAS plant, conclusion and reference. Let's start with introduction. A wound is considered a disruption of normal anatomical structure. Accordingly, healing is a very complicated and dynamic process that restores both anatomical continuity and function. The body typically responds to injury, infection, or other harmful stimuli that induce inflammation. It is an inflammation when a wound swells, gets red, and hurts. Jatropha CURCAS is a medicinal plant with multi-therapeutic properties like an anti-inflammatory, anti-malarial, anti-cancer, and antioxidant. What is wound healing? Wound healing is a complex multi-step process involving cellular and biochemical activities that start in response to injury, aiming to restore the function and integrity of damaged tissue. This process consists of continuous interactions between cells and their surrounding matrix, occurring in four overlapping phases. First, coagulation controls blood loss from damaged vessels. Second, inflammation takes about 0 to 3 days, the body's immune response to the injury. Third, reepithelialization 3 to 12 days involves the growth, movement, and development of skin cells to cover the wound. Fourth, collagen deposition and remodeling, which takes about 3 to 6 months, strengthens and reshapes the new tissues in the dermis. A network of blood cells, cytokines such as TNF-alpha and interleukins, growth factors like TGF-beta, VEGF, IGF-1, extracellular matrix proteins including integrin and fibronectin, and matrix melanoproteinases all work together, leading to tissue repair and regeneration. Wound healing treatments can be categorized into two main types: traditional therapies including herbal and animal-derived remedies, living organisms, silver-based products, and simple dressings, and the other modern therapies involving skin grafts, advanced dressings, bioengineered skin substitutes, and treatments using cells or growth factors. Each therapy type varies in effectiveness, clinical acceptance, and side effects. Herbal remedies are the most widely used traditional treatments for skin wounds because they are safe and effective. Scientific research has shown that herbs have multiple beneficial effects, including reducing bleeding, inflammation, and aiding in tissue repair activities like fibroblastia, epithelialization, collagen formation, and wound contraction. Faster wound healing lowers infection risk, complications, and cost, and due to the diversity of active compounds in plants, herbal products are promising agents for stimulating the healing process. What is inflammation? Inflammation is a complex biological reaction in blood vessels that occur in response to harmful stimuli such as injury, infection, environmental factors, cancer, or cellular changes. This response serves as the body's protective mechanism to eliminate harmful agents and kickstart tissue repair. The inflammatory process involves the activation of white blood cells, the release of immune chemicals like cytokines, and the production and release of inflammatory mediators and prostaglandins. Inflammation can either be acute or chronic depending on the progression of the condition. Acute inflammation typically presents with heat, redness, pain, swelling, and loss of function. Chronic inflammation, on the other hand, leads to gradual change in inflammatory cells and is marked by both the destruction and healing of the affected tissue. Here is a diagram of injury inflammation. Mechanism of anti-inflammatory activity. Early stage inflammation is essential in wound healing as it helps prevent microbial infections and remove dead cells and debris. This stage produces pro-inflammatory mediators including interleukins, TNF-alpha, inducible nitric oxide synthase, and chemokines, which are crucial for driving inflammation during wound repair. Research has shown that pro-inflammatory M1 type macrophages are more common in the early phases of repair, while anti-inflammatory M2 type macrophages, which aid in tissue repair, are more prominent in later stages of healing. As healing progresses, macrophages release anti-inflammatory mediators such as IL-1 receptor antagonists, decoy IL-1 receptor type 2 and IL-10, and growth factors such as TGF-beta, VEGF, and IGF-1, encouraging fibroblast growth, extracellular matrix production, and new blood vessels formations. In the final stages of wound healing, macrophages help manage ECM composition and remodeling by secreting proteases like matrix melanoproteinase and their inhibitors. MMPs breaks down key ECM molecules such as collagen, elastin, laminin, and fibronectin, and process cytokines and growth factors, ultimately leading to skin regeneration and scar formation. Herbal products with anti-inflammatory effects may support effective wound healing. In wounds treated with herbal products, there is a reduction in the buildup of leukocytes and pro-inflammatory mediators like interleukins and TNF-alpha. Studies show a marked decrease in TNF-alpha, CXCL1, and CCL2 levels, along with a reduction in neutrophil and macrophage accumulation. Research also indicates that the transition of macrophages from the M1 to the M2 phenotype is crucial for effective healing. It is well established that TGF-beta released by macrophages stimulates the transformation of certain fibroblasts into myofibroblasts and enhances collagen formation production, leading to ECM contraction and eventual wound closure. Additionally, TGF-beta 1 and TGF-beta 2 encourages scar formation, while TGF-beta 3 may help minimize scarring. Jatropha curcas plant. Jatropha curcas is a plant belonging to the Euphorbiaceae family and the genus Jatropha. Native to the Americas, this plant is widely distributed and also found in Africa, Asia, and Europe, where it thrives in arid tropical and subtropical regions. Jatropha has over 200 names across different countries. Some common names include purging tree, curcas nut, and psychic nut, and in Hindi it is known as Ratanjot. Distribution: India, found in Tiruchirappalli, Pudukottai, Tanjavur, and Ramanathapuram districts in Tamil Nadu, as well as in Pondicherry. West Africa, including Nigeria, grows as a weed in crop fields, bush report areas, roadsides, and distributed areas, especially in regions with higher rainfall. The name Jatropha comes from the Greek word Jatros, means doctor, and trop means food, reflecting in traditional medicinal uses. The medicinal value of Jatropha curcas is well documented, and various parts of the plant consumed in different forms such as infusions to treat conditions like fever, burns, rheumatism, allergies, diarrhea, intestinal cramps, wounds, dysentery, and parasitic infections. Preclinical studies have confirmed the biological activity of various parts of Jatropha curcas. The seed shows antifungal and antiarrhycidal effects, while the root, stem, bark, and leaves have antidiarrheal, anti-inflammatory, and antimicrobial properties. These therapeutic effects of Jatropha curcas are likely due to its secondary metabolites such as alkaloids, flavonoids, esters, lectins, tannins, saponins, terpenoids, and lipases. The classification of Jatropha curcas plant is as follows: Kingdom Plantae, Order Malpighiaceae, Family Euphorbiaceae, Genus Jatropha, Species J. curcas. The diagram of Jatropha curcas. Table of content of Jatropha curcas with various parts like leaves, stem, bark, latex, seeds, and roots. Advantages of Jatropha curcas plant. The Jatropha curcas offers various benefits for wound healing, including speed up healing, strengthens skin and granulation tissue, enhances wound contraction, boosts hydroproline levels, reduces epithelialization time, and promotes collagen production. Disadvantages includes toxicity. Every part of the plant is poisonous, with the seeds being particularly hazardous due to a high concentration of resin. Ingesting the seeds can lead to symptoms such as vomiting, diarrhea, abdominal pain, and a burning sensation in the throat. Phytotoxin. The plant contains a phytotoxin known as curcin, which may lead to dehydration, cardiovascular collapse, and central nervous system depression. Forbolisters. The seeds also contain forbolisters, which are toxins that cannot be neutralized by heat. However, they can be converted into less toxic compounds using water or ethanol. Conclusion. Herbal products have been used for skin wound care for a long time. Numerous preclinical in vivo studies on animals, along with a few clinical trials, have demonstrated the effectiveness of herbal products in promoting wound healing. This study suggests that the aqueous extract of J. curcas leaves has an anti-inflammatory effect comparable to standard drugs like ibuprofen. The anti-inflammatory activity of the aqueous extract indicates its potential influence on the synthesis of kinins, prostaglandins, bradykins, and lysozymes. Alkaloids and flavonoids found in plants are known to have significant anti-inflammatory properties, and phytochemical analysis of J. curcas has confirmed the presence of these compounds. The study showed that the crude bark extract of J. curcas effectively accelerates the wound healing process in Lister albino rats. It also highlights the wound healing properties of the methanol leaf extract using incision and excision models in rats. At a dose of 200 mg per kg, the extract demonstrated significant healing potential when compared to the standard cetirizine, showing a dose reduction in the epithelialization period. Here are some references. Thank you.